It's going ahead.
All right. Okay, I don't see it appearing here. So I'd like to welcome you to our first talk for 2026 for the IEEE Magnetic Society for the Twin Cities. We hope to have a very interesting roster of speakers coming along in the next couple of months through. Just as a general kind of introduction to the IEEE for those who are not familiar. So its mission is to foster technology, innovation, and excellence for the benefit of humanity. And the vision is to be essential to the global technical community and to technical professionals everywhere. I would mention that it is the largest global volunteer professional and technical nonprofit worldwide. Next in the rank order is, I guess, so medical and realtors in terms of the organizations that are out there. So I'm very excited to introduce our first speaker this year. It's Dr. Kai Wu. He's been from the Twin Cities, but he's a tenure-track professor in the Department of Electrical and Computer Engineering at Texas Tech University. He got his PhD in electrical engineer here at the University of Minnesota in 2017 and continued to work as a postdoc here until recently, 2022. His research is focusing on integrating magnetic nanomaterials and spintronic devices to advance biomedical diagnostics and therapy. With that, I would like to hand it over to Dr. Kai Wu.
Thank you, Chris, for the introduction. Let me share my screen here. All right, assume everyone can see my screen and can hear me, right?
Yes.
Okay, great. Yeah.
Okay, sounds great. So yes, my name is Kai Wu, as introduced by Chris, and I was a student in Dr. Jinping Wang's lab until I joined Texas Tech in 2022. So today, also as introduced in my, let me get that pointer. Okay, as introduced in the abstracts, like I have been working on the magnetic material and spintronic devices for biomedical applications. However, today's talk, I will only focus on the magnetic nanoparticles for biomedical applications, for biosensing and imaging application. All right, without delay, I can continue with my talk here. Okay, I will first introduce about my previous training and also my current lab's focus on research areas. So I spent like 9 years in Dr. Jinping Wang's lab for my PhD and postdoc. So I summarized my previous research in 5 topics. And as all of you have heard about the GMR-based biosensing, I was one of the team members at that time from 2013. So until Dr. Wang has the startup company Zap2 Life in 2015. So I was involved in the application, including the chemical surface and treating on the GMR sensor surface to be able to functionize with antibodies or DNAs for the specific bonding of the virus and some proteins. You can see some examples that we have used the GMR POC device for the cardiovascular biomarker detection, the influenza virus H1N1, H3N2, and ovarian cancer testing. Most recently, in 2022, we applied for the testing of the whole tumor cell, OSCA8 tumor cell. In the same time, during my PhD, I also independently built up the magnetic particle spectroscopy we call MPS POC device, which we are using the relaxation dynamics of nanoparticles for biosensing applications. What we do is like we functionize the nanoparticle surface with proteins or antibodies or DNAs that they can cross-link with other MPs when there's an object or analyte in the sample. So this cross-linking process, the bonding process will delay the realization process of these nanoparticles, which we can detect the signals from particles to give out quantitative data of the concentration or amount of biomarkers in the sample. So I will introduce this topic in today's talk as well. The third one is we are trying to implant the micro device, nano device into the brain for the neural stimulation. So this was in collaboration with Mayo Clinic that initially we thought about using the spintronic device to stimulate the cells in the brain. However, it did not went very well, so we moved to the microcoil-based stimulation, and successfully we demonstrate the microcoil-based neural stimulation in the hippocampal part of the rat, and also the forebrain bundle FBS bundles in the rat, which trigger the muscle contraction and also trigger the release of dopamine in the brain. So this point of one way of that we can use the magnetic micro device for the treatment of some brain disorder in the human or in animals as well. The fourth topic I did was the synthesis and functionalization of the iron nitride particle. As you also know that the Niron company is so popular nowadays, right? My focus is like how we can transit this iron nitride powder into nanoparticles, and we functionalize them for the biomedical applications. So this one topic I was working on, that I successfully functionalized the surface of the iron nitride nanoparticle with some polymers to increase the stability in the biofluid sample for the biomedical applications. The last one I developed in 2019 is micromagnetic modeling that uses mu-max or OMF to the modeling of the device, such as spintronic devices, which all of you know that is very popular tool site for modeling the device part, the performance of the device. So in summary, the 9 years of training at Minnesota, I covered the spintronic sensors, immunoassays, biomedical application, point-of-care device like the tool device we built up in the past 10 years, and the nanoparticles and the modeling part of the device. So now I want to talk about the independent work in my lab. Since 2022, I joined Texas Tech. So I continue working on the biomedical application, but I mostly focus on the nanoparticle-based applications. As I'm going to present in today's talk, we can synthesize and functionalize nanoparticles with varying size, shape, and element doping to change the magnetic properties of these particles for different applications. So this project is funded by NIH R15 and R16 grant that initially we plan to do the hyperthermia treatment of tumor and also for the separation of the blood cells in the human blood, as well as the biomedical imaging. The second topic, as an EC background training, I also try to build up an imaging facility that is benchtop, and we try to build up the magnetic particle imaging, which is very similar to the MPS, but we do the three-dimensional in the whole space imaging to do the scanning, to imaging the distribution of nanoparticles in the tumor region. So I will also spend some slides to talk about this topic in my current lab. This project is funded by the NIH R03 grant. And as you also know, that very popular topic nowadays in memory computing, right? As a biomedical engineer, I also think about how we can learn about in-memory computing to apply for biomedical applications. So what I'm working on currently is in sensor computing. We want to build up hundreds to thousands of the spintronic sensors to detect the brain signals from the brain. So with such a large amount of data the data bus passing through the thousands of hundreds of sensors, we want to reduce the data processing time and also lower down the power consumption for the own sensor computing. So what we do is like we borrow the idea from the in-memory computing, and we want to build up the spintronic device for the brain imaging, and we want to involve the in-sensor computing to lower down the computing power consumption. So this is one wearable application that we are trying to do in my lab as well. The fourth topic is 2D materials, which is a very new and fundamental topic I'm working on. So currently I'm thinking about the 2D material for biosensing applications, as I know that Dr. Chen Gong from Maryland, he is trying to work on the 2D biosensors as well, that we have to hurry up on this 2D material, how to apply it to biosensing applications, how to make the surface of this thin film structure to be stable, to interface with the biological environment is the challenging part. And how to make the result or data repeatable in the biological environment is also one challenge we want to solve in current stage. The last one is AI for healthcare. As you also know that I work on the biomedical imaging. So with imaging, we can produce a lot of data. And we are trying to involve AI to analyze data to lower down the burden on human beings. So one application I showcase here is I collaborate with a doctor from the TTU Health Science Center that we want to use AI to analyze the skin burn area of the body. So this is a very interesting topic for the US Army that for the soldiers, if they have the skin burn, we can use AI. We can use the cell phone camera to take a photo and AI algorithm to categorize the burn degree and the total burn area to guide the treatment of drugs on this burn area. So these 5 topics are the new research areas I developed in my lab since 2022. In summary, the keywords covers spintronic sensors, 2D materials, nanoparticles, magnetic imaging, in-center computing, and AI or machine learning area. Okay, so long I introduce my background and I tell you my capability or my lab's capability. Now I want to focus on the topics currently in my lab, nanoparticles. So my lab can produce nanoparticle in different methods like a co-precipitation method and hydrothermal method I present in today's talk. So in the co-precipitation method, we are using the ions as the precursors, and we can dope the divalent ions nitrates into it. For example, you know the iron oxide Fe3O4 nanoparticles. We can dope the divalent elements such as zinc, manganese, and cobalt into these nanoparticles to change the anisotropy and MS values of these particles to vary their realization dynamics for biomedical applications. So the fundamental part is like we are doing some doping on this iron oxide to change the dynamics of these particles to apply for hyperthermia treatment, magnetic particle imaging, or biosensing applications. So this just like we do a small change and the outcome on the application will vary a lot. So in the co-precipitation, as you can see, we can generate a large amount of particles, but the particle shape and size is difficult to control. As you can see, we try to produce cubic and spherical particles, but they are not very perfect in the shape as we want. So we also compare with the hydrothermal method to produce nanoparticles. This method is better at controlling the size, uniformity of particles, and also controlling the shape of particles. We showcase the spherical particles produced by hydrothermal process. And also we showcase one example, one exception. We also produce cubic iron oxide with a very large size to compare the performance with the particles we produce here. So overall, in this topic, I'm going to present to you the calibration of these lab-produced particles. We have 5 cubic particles with different doping, zinc manganese doping, and cobalt doping, around 10 nanometer in size. We also have 4 spherical particles produced by hydrothermal method with around 10 nanometer size, 10 to 12 nanometer size. And we also have the zinc manganese, and cobalt doping. One exception with the cubic nanoparticle with 35 nanometer for comparison. So today's talk, I will cover the calibration of these 9 particles in the physical properties and the biomedical properties as well. So we first calibrate the crystal structure of these particles. So I compare the cubic nanoparticles and spherical particles in 2 panels here. So we mark the XRD plans of the iron oxide plans here. As we zoom in into the 311 plan, you can see that in the 311 plan of the peak, you can observe the peak shift as we do some doping like a cobalt, zinc manganese into the iron oxide. This peak shift, as evidenced by the lattice parameter of these particles, you can see that with doping of element, the lattice parameter will increase compared to the iron oxide the initial phase Fe3O4, which tells us that we successfully doped this element, divalent element, into iron oxide, into iron oxide to achieve different doped ferrous structures. The same thing you can see from the spherical particles. We also see the peak shift of the 311 plane. And also we observe the increased lattice parameter after doping. Okay, next we measure the image curve of these particles. We also compare with the 35 nanometer Fe304. As you can tell from the sides, the 35 nanometer Fe304, they are not superparamagnetic. So you can see a very large hysteresis loop from this Fe304, 35 nanometer with the red curve. For the other particles, the coercivity is almost zero. So we claim that they are superpara and they can be used for biomedical applications. So the top panel shows you the calibration of different cubic shape MMPs and spherical shape MMPs. If we cross compare with these two different shapes, you can see that all the particles show a saturation magnetization around 55 to 75 EMU per gram, which is better than the particle we can buy from the companies. Secondly, we also observe that cubic MMPs with induced shape anisotropy, they show higher MS values compared to the spherical particle of the same size. And you can compare with these two figures here. Okay, just a slightly better. The cubic MMPs shows a slightly better higher MS values compared to the spherical particles of the same size. All the particles are superpara when we see the image curve here. Next, we calibrate the cell viability or the biocompatibility of these particles into the SKOV3 ovarian cancer cells. We incubate two different concentrations of 100 microgram per milliliter and 500 microgram per milliliter of particles into the ovarian cancer cells. And as we see from all the figures here, the cells are highlighted by the green color. You also can see some red color of cells, very minor amount, as you can see from the laser point here. So the red color shows that the cell is dead. The green color shows that the cell is still alive. So based on all the 9 particles we tested at 2 concentrations, we can confirm it has over 99% of the cell viability in this testing over 24 hours of incubation. And which tells us that the negatively charged MMP surface, they generate a minimal ROS, which is the reactive oxygen species that lower down the cytotoxicity of these particles to the cells. Okay, so for all the particles we produce, they show very high biocompatibility with the incubation with the cells. We also want to see if we will apply these particles for biomedical imaging. If, for example, if you want to imaging the tumor cells, we want the tumor cells to uptake the nanoparticles into the cell membrane. So this will be better showcase that the particles distribution in the tumor cells, in the tumor regions. So the next stage, we want to test the cellular uptake of these nanoparticles. We incubate these particles with the SKOV3 cells. And among all these images, you can see that some particles like FeO3 cubic nanoparticle, they show some uptake of MMPs inside the tumor cell. You can see from the boundaries of the cell membrane. All the figures, you can see some very light color of the boundary of the cell membrane, and you can see the particles, the darker color, they are accumulating inside the tumor cells. So which shows you that all the particles has some various level of cellular uptake. I also want to highlight 3 particles. As you can see, I highlighted them, the zinc manganese doped cubic particle and the cobalt doped cubic particle, as well as the 35 nanometer Fe304 cubic particles. They show much higher cellular uptake of the nanoparticles, which tells you that if we want to choose particles for imaging purpose or the hyperthermia treatment of tumor, we want particles to be internalized by the cells. So the better choice will be these 3 particles. They will increase the amount of particles that internalize in the cell and create a higher signal or better performance in hyperthermia treatment. Okay. The next, we also want to demonstrate and these particles used for the tumor treatment by hyperthermia process. So we prepare the particle into an FBS supplemented SKOV3 cell membrane. FBS is fetal bovine serum. It's one typically used biological buffers. We supplement it with the SKOV3 cells in the media. Then we incubate these particles with this liquid medium on the top figure. We also incubate this particle into an agar phantom embedded with the tumor cells. This agar phantom is a mimic of the human tissue, which is a solid structure, as you can see from the photo here. So in the agar phantom, the Brownian motion of the nanoparticles will be blocked. So the hyperthermia heating will be produced by the hysteresis loss of the particles through the Near process. When the particles are in the liquid as the top one, they are in the FBS buffer liquid with the cells. They can generate heat through the joint hysteresis loss and also Brownian and Near process. So you can tell that the heating, when the particles are in the liquid, they will heat up the sample very fast. The delta T temperature increase to 40 degrees C in short time. However, when they are embedded in an agar phantom, the Brownian motion is blocked. The heating can only reach to 25 degrees C. So in any case, we only need the particles to heat up 10 degrees C to kill the cancer cells in the body. So all the conditions show us that the particles are very good candidates for hyperthermia treatment in vivo and in vitro application as well. I also want to mention that the AC magnetic field we apply to the hyperthermia treatment is within the clinical safety for the human body, which is 30 millitesla and 101 kilohertz, which is safe for the human body in vivo use. Here's a photo of the system we use to calibrate the hyperthermia performance of these particles. And after the treatment, we also want to test if the tumor cells are really dead or killed by the hyperthermia process. So we did the green-blue test again, that as you remember, the green colors dots represent a cell is alive. The red dots represent a dead cells. So based on this performance, you can see that most of the cells, most of the cancer cells are killed by the hyperthermia process. This is because the MMPs, they produce the heating, nanoscale heating, which will disrupt the cell membranes. And also this heating will improve the reactive oxygen species generation, which will also disrupt the membrane in the mitochondria, the cell organs inside the cells. As you remember, the tumor cells, they will internalize the particles inside. So the nanoparticles will directly interact with mitochondria or the organelle in the cell. This ROS generation will kill or disrupt the potential and the membrane of the mitochondria in the cells, which will also cause the cell death. So this joint phenomena of heating and ROS generation is the major drive force to trigger the cell death in the hyperthermia treatment. The next one, we want to say that for most biomedical applications, many people or many papers I read, they ignore one fact. They inject the nanoparticle into the blood vein. The particle will first interact with the human blood cell. People rarely consider how the human blood cell will accept the nanoparticles in the blood vein. So we did the hemolysis test that we incubate these nanoparticles with human blood cell we collect from the donors from the HSC, the TTO Health Science Center. We vary the concentration of the particles from 200 microgram per milliliter to 1000 microgram per milliliter. So this low concentration mimic the intravenous administration of particles into the blood vein. The 1000 microgram per milliliter mimic we inject the particle directly into the tumor, which we call intratumoral administration of particles. And you can see that for the zinc manganese doped cubic particles with 10 nanometer size, the human blood cell, we do not see blood cell deformation of the shape or aggregation of blood cells after 1 to 24 hours of incubation. This confirms that this nanoparticle will not generate any mechanical or magnetic stress to the red blood cell membrane, which is safe for the intravenous or intratumoral injection. As a comparison, we compare the negative control group of AS3's buffer. This is buffer to storage the human blood cells in the blood bank. So that you can see that blood cells are very good in shape and no aggregation. If we also compare with the PBS buffer, salt buffer people typically use in the lab, you can see the blood cell membrane has a shape change, a spikes change on the membrane, which means the membrane deformed due to the interfacing of the blood cells with the buffer. Although the particles, you can see that the image is like so dirty, right? These are the nanoparticles with this blood cell. But if you focus on the blood cells, the lighter color, light gray color, the blood cells are very intact and they are very good shape. So this further demonstrates that this nanoparticle is really well compatible with the human blood cells. As a comparison, we also compare the iron oxide cubic particle with 35 nanometer, very large cubic particles. You can see that this particle first will introduce nanoparticle aggregations, very severe, the RBC and the blood cell aggregation. In addition, it will also disrupt the membrane of the cells. You can see some spikes on the surface of the red blood cells, which tells you that these nanoparticle, large cubic Fe3O4 nanoparticle, they are not well compatible with the human health, with the human blood. So this tells us that this particle cannot be used for in vivo applications, such as injection to the human blood veins. So lastly, we already show you that some particles are biocompatible and some particles are good for hyperthermia therapy for the tumor treatment. Now I want to further demonstrate that this particle can also be used for the tumor imaging in MRI system. The full name is magnetic resonance imaging. So we prepared some phantoms of these nanoparticles embedded with agar phantoms with SKOV3 cells. The top panel shows zinc manganese doped cubic particle with 10 nanometer size. The bottom panel shows the MRI imaging of the iron oxide cubic particle with 35 nanometer. You can clearly see that for the bottom panel, the imaging of the MRI imaging is not very smooth. You can see some bright color, some dark color, right? This not very smooth of MRI imaging, we call it as an artifact. So which tells you that the 35 nanometer cubic iron oxide nanoparticle, they are creating some sharp images in the MRI imaging. So these are not ideal for in vivo application because they act as an artifact. You can see that some very bright color and some very dark color of the nanoparticle in MRI imaging. They can create some islands of the tracers or islands of hyperthermia treatment, islands of hyperthermia heating, which is not very good for clinical applications. On the other side, for the 10 nanometer zinc manganese doped MMPs, the imaging are very smooth compared to the bottom one. The MRI imaging is very smooth, which gives you a very smooth or evenly distributed particles in the space, which is ideal case for the MRI imaging and hyperthermia treatment. So so here I showed you that these particles are capable for the MRI imaging and also capable for the hyperthermia treatment. So last one, as we proceed for the last step, as we proceed for in vivo application, we want to see if these particles are real compatible, if there's a perfusible blood vessels. What we do is like we do the 3D bioprinting of the SKOV ovarian cancer cells with hydrogels. As you can see from the figures on the left side, after bioprinting, we can create a perfusible blood vessels, the vessels with a diameter here. As you can see, we can pass the nanoparticles through vessels to mimic the nanoparticles passing through the blood vessels in the human body. So this particle will penetrate into the gelma or to interact with the cancer cells. So after we passing through the particles into the perfusible blood vessels for 48 hours, we can still see that most of the cancer cells are still alive from the color maps here. So this tells you that even the nanoparticles are circulating in the blood vessels in the body for 48 hours. It is still biocompatible with the human cells. So in summary of this topic, we showcase the capability in my lab that we can synthesize and fine-tune doping nanoparticles with different size and shape. We also can use these particles for in vivo and in vitro application of the hyperthermia therapy of the tumor cells and for MRI imaging. And we also can do the 3D bioprinting to print some biological phantoms to demonstrate this performance of particles before we move to the in vivo application. All right, so this summarizes the topic of number 1 in my lab. And before I want to talk about the magnetic particle imaging, the tumor imaging, I want to briefly recap the MPS topic I did when I was a student in Minnesota. So this part, number 3, is talking about the MPS device, how we build up from the prop station to a POC device and to commercial product. So I'm the students that first initialized this topic in Dr. Wang's lab in 2014. At that time, I used a benchtop system, that very simple system. As you can see that this system I built up to demonstrate the blood hyperviscosity testing on this MPS system. We also demonstrate the multiplex biosensing on MPS platform. We also filed some patents, a total of like 5 patents on the device design and bioassay theory design as well. In 2019, I led a team of over 23 students, including graduate and undergraduate students, to build up the first POC device in 2021. So we applied this device for the detection of the H1N1 influenza A virus, SARS-CoV-2 virus as well. And in 2024, last year, Dr. Wang built up the company based on this technology. So how does the MPS works? Most of you probably know the GMR biosensing, right? The GMR biosensing is a surface-based biosensing. We need a sensor to convert the nanoparticle signal into the readable voltage signal that we can read from the GMR sensor. However, MPS, they get rid of the GMR sensor, which will tremendously lower the cost per assay. We are using nanoparticles as the signal source and also use it as the props for biosensing application. What we do is like we can functionalize particle with antibodies through a chemical covalent bonding process. This antibody will bond with epitopes from the proteins or virus or bacteria. So this bonding process will cause the clustering of nanoparticles into one analyte or one virus or one pathogen. This clustering will block the Brownian motion of particles. Imagine you have nanoparticles. You apply an AC magnetic field to switch up and down. So the particle will try to realign the magnetic moment of particle to the field through the joint Neal and Brownian process, as I show on the left figure. After the cross-linking of particle in the presence of the analyte, the clusters will block the Brownian motion of particles, which will reduce or lower down the dynamic response of particles to the magnetic fields. So what we observe is we showcase a very simple structure that we apply the 2 AC magnetic field. And we also have the image curve of the silver paramagnetic nanoparticles. The time domain response of particles magnetization or magnetic moment will be modulated by the AC magnetic field. So this moderation process will generate higher harmonics that does not belong to the field we apply. It belongs to the particles dynamics process. So we compare the higher harmonics produced by nanoparticles compared to the field, the FFT, the frequency domain of the field we apply. So these harmonics are the signal we use to calibrate or quantify the dynamics of nanoparticles in the case of the amount of analyte in the solution. So usually we are using the Brownian motion dominated nanoparticles, which means the signal mainly comes from the Brownian motion of particles. However, if the Brownian motion is blocked, we will see a drop on the harmonic signals in the amplitude. So the drop of the amplitude of harmonics is proportional to the amount or concentration of the analyte in the solution. So the whole story, the logic part is like nanoparticles will form clusters in the presence of analyte. So this will hinder the Brownian motion, will lower down the dynamic response of particles to the magnetic fields, so which will lower the harmonic signal we observe. Okay, so this is the whole story about how the MPS works to be a quantitative biosensor platform. Here shows you the photograph of the POC device we developed in 2021. As you can see that on the circuit board, we have the microcontroller to control the generation of the sinusoidal wave, one with the high frequency FH, one with the low frequency FL. So this high and low frequency sinusoidal wave, they will drive the coils to generate the AC magnetic field. This field will apply to the nanoparticle as we put in the glass vial in the center part. So the nanoparticle signal will be collected by a gradiometer. This gradiometer will convert the time domain magnetic signal, will convert to the voltage in the coil following the Faraday's law. So after we get the voltage from this pickup coil, we do the amplifier, filtering, and ADC conversion to get the frequency domain of the signal of the harmonic amplitude. So we showcase two different IC platforms. One is we apply a single drive field with frequency F to the nanoparticles. We will get the higher harmonics at 3F, 5F, 7F, these odd harmonics from the particle's response. Or we can do the two frequency combination, one with high frequency, one with low frequency. This frequency modulation will moderate the higher harmonics around the FH components, which is usually 5 kHz. So we can collect the harmonics from FH plus minus 2FL as the third harmonic, FH plus minus 4FL as the fifth harmonics from the nanoparticles response. So these slides shows you the POC device we built up and the circuitry that from the signal generation to signal collection from the nanoparticles. We also built up a user interface. I think it's still available on Google Play that you can download for free. The user interface will guide you to load the sample into the vial and put the sample into the POC device and wait for 1 second or 5 minutes to incubation to get a result from the user interface. So it will lower down the cost compared to the GMR biosensing device. So we estimate each MPS POC device can cost less than $100, and each testing vial only costs you $1 to $3 for the MMPs and antibodies we use in the nanoparticles. And the application is free to download. So the advantage compared to the GMR biosensing is like all the assay happens in the solution. We call it a volumetric-based biosensing. So nanoparticles can move freely in the solution and bond with analytes, forming the clusters. So this will lower down the bioassay time to 5 minutes compared to the typical biosensing platform. Okay, so here shows you one result I collected in 2020 that we test this MPS device with varying concentration of the H1N1 virus. As we vary the concentration from very high concentration, the black curve, high concentration of the virus proteins with low concentration, zero concentration of the proteins, or maybe only nanoparticles with no antibody coatings. You can clearly see that with higher concentration of the analytes, the harmonic signal will drop due to the bonding causing clusters, which will lower the dynamic response of particle, which will lower the harmonic amplitude of the third harmonic amplitude. We can also see from the TEM imaging of the after the MPA assay, you can see the proteins will cause the clusterings of nanoparticles when there's a high concentration, 2.2 micromolar concentration of the proteins in the solution. With a lower concentration of the proteins, 88 nanomolar concentration of the proteins, the cluster is less severe from the TEM image. When there's no protein, the nanoparticles barely cluster in the TEM image, as you can see. So this further demonstrates that higher analyte concentration will increase the degree of the MMP clustering, which will lower the amplitude of the harmonics you can collect from the MPS device. Lastly, the last part I want to introduce is in the pandemic, we got some funding from the NIH that we want to test on SARS-CoV-2 detection on the MPS platform. So we focus on the spike protein and the nuclear protein from the SARS-CoV-2 virus. This schematic image shows you that the spike protein are like the spikes. They are crossing the membrane of the virus. The nuclear protein is a protein that's wrapped with the RNA strands of the virus inside the viral particles. So we demonstrate the MPS to detect both spike protein and the nuclear protein from the SARS-CoV-2 virus in this work. So what we do is the first time we introduce 5 minutes bioassays on MPS bioassay, MPS platform, which is the first report in the world that we can cut down the bioassay time from the sample loading to data reading. As you can see from the schematic imaging, we functionalize nanoparticle with the antibodies. So this will be the product as I highlighted in the step number 2. This product will be the one the user will get. The user will get a small vial of nanoparticles, which is functionalized with antibodies. The user will add the samples, biofluid sample, into the nanoparticle vial. In a typical process, people have to wait for overnight for the bonding to happen, right? So in our case, we did a shortcut. We apply the mixture of nanoparticle with the biofluid sample under the agitation and heating process. So this mechanical agitation and heat and also thermal heating process will speed up the Brownian motion of particles. The particles will move more frequently. This will increase the bonding chance of the particle to bond with analytes to form clusters. So this process tremendously cuts down the incubation time from 1 hour to 5 minutes. So as we anticipate, the analyte will cause the clustering of the nanoparticles, will lower down the harmonic amplitude. You can see from the figures on the right side, as we summarize, as we increase the concentration of the SARS-CoV-2 spike protein concentration, you can see a drop of the harmonic amplitude forming the almost linear curve or S shape. So this gives you the detection range of this biosensing platform from sub 1 nanomolar to over 1 micromolar, and also a linear range of detection of this biosensing platform. So overall, at the end of this topic, I want to summarize that we demonstrate the MPS POC device for detection of the MMPs, that when they forming clusters, we can detect quantitatively about the analyte concentration and amount. Secondly, we cut down the incubation time from 1 hour to 5 minutes by introducing the agitation and heating process in the incubation process. And we demonstrate the biosensing for SARS-CoV-2 and influenza A virus detection. All right, the last part of this talk will be the imaging. the reason I want to introduce MPS biosensing because the MPI imaging is based on MPS sensing sensing idea. So my lab is currently building up the MPI imaging platform that we can do the 3D scanning of the tumor, which is marked by the nanoparticle tracers. So what we do is like we are using the DC magnetic field to create a field-free point. Imagine in the whole space, two magnets are facing each other. They will create a gradient field. In the center point of the space will be a field-free point, which means the DC magnetic field is 0. Outside of this point, there will be a non-zero DC field we call gradient field. So if you can recall the S shape of the silver paramagnetic nanoparticles, when there's a DC shift, DC field to the particle will be fully saturated by the DC field, non-zero DC field. So at this time, the particle is locked. When you apply the AC magnetic field, the particle will not generate any change of the magnetization upon the AC field because the particle is fully saturated by the DC gradient field. Only the particle in the field-free point, in the center of field, they can still be responsible or generate a response to the AC magnetic field. So the particle inside the field-free point, they are like in MPS system. They will generate the higher harmonics in response of the AC field. But the particle outside the field-free point, they are fully saturated by the DC field, non-zero DC field. They cannot generate any signal into the system. So in this way, if we can program by programming a focus field and a drive field, we can move the field-free point to scan the whole space. Like if you're on the beach, you can use a metal detector to scan the surface line by line. The same thing, we are controlling the focus and the drive field to scan the FFP, the field-free point, to scan the whole space, 3D space. And at each data point, at each time, only the particles, they are inside the field-free point, they will generate a signal. So in this way, you can scan the whole space to recreate a 3D tomographic imaging of the distribution of nanoparticles. If these particles are internalized by the tumor cells, you will see the distribution of the nanoparticles is equal to the 3D image of the tumor cells, tumor tissue. So that's how we can realize the tomographic imaging on MPI system. I also show you the field parameters of the drive field, AC field, with 20 kilohertz AC field. We also have the focus field that can move to help move the AC field, to move the field-free point to scan the whole space. We also have the selection field that will generate the gradient field that will create a field-free point in the one small space in the scanning space. So this platform we want to build up has a scanning space of around 2 by 2 by 2 centimeter cube field of view. So we can put an object of this size into the MPI system to do the 3D scanning. All right, the next we want to calibrate if these coils, because you understand that we have so many coils, the gradient field coil, the focus field coil, XYZ direction, YZ direction focus field, and also the drive field coil in the X direction. All these coils, when they generate a magnetic field, they will interfere with each other. So we first want to demonstrate if these fields are uniform in the field of view or in the scanning space. So this figure shows the finite element modeling of the field profile of this drive field, 20 kilohertz, selection field, DC field, and Z and Y focus field. These are the slowly change field to switch the location of the field-free point in the space. So you can see almost 95% field uniformity in the field of view, 2 centimeter field of view. So this tells us that this coil design with dimensions we entered here are able to generate a uniform field for 3D scanning in the field of view we want to scan. Okay, Second, we do some modeling to see how to program the focus field to make sure that the field-free point can scan as we program to follow the trajectory we want it to follow. So we did a very small modeling that we control, as you can see, the three-dimensional X, Y, Z direction. We have the drive field along X direction. It can move the field-free point along the X direction. We also need the Y and Z focus field to move the scanning trajectory from one line scanning and move to the second line, the third line, then move to the other plan, plan 0, 1 line, 2 line, 3 line, plan I, the minus plan, 1 line, 2 line, 3 line. This is just a demonstration of how the focus field programming can guide the trajectory of the field-free point to realize a programmed 3D imaging. And I want to show the point. How do I change back to the highlight? Okay, so here the video shows you that we are doing the line scanning on the XE plane, the plane facing you. We do the 3-line scanning to control the purple color is the field-free point, which means at each time point, only nanoparticles inside the purple color region, they can generate the harmonic signal. We can use it for image reconstruction. We also show you the plan by plan scanning, 3 planes scanning in the Y direction, 3 planes. So this demonstrates that we can control the magnetic field to program the scanning of the MPI in the whole 3D space. Besides that, my lab also doing the micromagnetic modeling of the dynamics of particles under the AC magnetic field. Some of you might work on the dynamics of nanoparticles under high frequency field. So in this case, the typical LLG equation does not fit at all, right? So here we are using the stochastic Landry model, which combines the typical LLG equation with the Brownian motion equation on the bottom row here. So we label the magnetic moment vector as a unit vector M, and the axis or the crystal anisotropy axis N as the vector N here. So we are doing the iteration of these 2 vectors under time varying magnetic field C here. So the C contains the time varying magnetic field and also the anisotropy equivalent field. And we also consider the thermal energy at a finite temperature. We convert to thermal equivalent magnetic field, HTH. So based on this, we also involve the thermal noise into the nanoparticle system. We can accurately estimate the dynamic response of the nanoparticles in suspension solution. We can plot the real-time dynamic magnetization of these particles for imaging or MPS bioassay applications. We can also get the AC image loop of particles. We can also plot the higher harmonics and point spread functions that is guiding the resolution and spatial resolution of the MPI imaging. This slide shows you that some data we collected by modeling the dynamics of nanoparticles under the AC magnetic field in MPI applications. So this will guide us in the nanoparticle tracer design. As you can see that in the time domain, the response of nanoparticles always has a phase delay to the AC magnetic field you apply. This is because the Brownian and Near process will cause a delay of the response of particles to the AC magnetic field. We can also reproduce the AC emission loop of the particles under this AC field based on the time domain emission curve, MT curve, as well as the higher harmonics of particles under one frequency, one AC magnetic field, 3F, 5F, 7F. All these harmonics will be used for the image reconstruction for MPI imaging, as well as the signal to noise ratio and also the point spread function, which is related to the spatial resolution of MPI imaging. So with the consideration of the time, I will just quickly go through these findings. We found out that at a lower excitation field amplitude, it will actually improve the spatial resolution of MPI imaging, but it will lower the signal-to-noise ratio for imaging. So in the other work, we also found out that larger particles can improve the MPI spatial resolution based on the steeper image curve in the ACMH loop. And if we have the MPs with higher anisotropy, it will also increase the imaging resolution. So one topic the UC Berkeley and the NIST group they are working on, they are using the nanoparticle chains, MMP chains for images. This is also introducing the shape anisotropy to improve the resolution on MPI imaging. We also consider the dipolar interaction between nanoparticles that they may affect imaging qualities. So we compare the MMPs with this is size. It's not dipolar interaction. Okay, so this slide about the MMP tracer size that we vary the MMP diameters from small size to the large size, and also we consider the particles are not uniform in size. They have follow a log normal distribution of the nanoparticle size. So with considering the particle size and the size distribution, we found out that larger nanoparticles will improve the SNR and the spatial resolution in MPI imaging, as we covered in previous slides. If the particle is not uniform, a broader size distribution, surprisingly, it will increase SNR, but it will degrade the spatial resolution of the imaging. This is because if the particle has a broader size distribution, the larger particle, their signal will dominate. They will create a larger signal compared to the smaller particles. So this is something that should be considered when people are using nanoparticles for imaging application. Okay. And we also found that surface coating with polymers or antibodies, this will have not much effect on the MPI performance. Lastly, I think this will be the last part. We consider about the dipolar interaction, which is very popular consideration in all industry and the biomedical application areas. If the particles are too dense to each other, they will interact with each other. So we consider this dipolar interaction of particles, and we vary the interparticle distance from small distance, very high dipolar interaction, to the large distance, no dipolar interaction at all with this 250 nanometer distance. And you can see from the signal, harmonic signal, and also the time domain response of particles. With the dipolar interaction included, the response of particle is weaker compared to the no dipolar interaction with the darker orange color, no dipolar interaction, with lighter color, high dipolar interaction. The dipolar interaction will reduce the response of particles under the magnetic field. It will also reduce MPI signal intensity, so which indicate that if the particles are aggregating in the tumor region, the signal is expected to lower down due to the dipolar interactions. Okay, yeah, so these are the research topic I want to cover. Besides this, I want to also mention that my lab also have an MPS system. We can use MPS system to calibrate the hyperthermia performance, the harmonics to calibrate for MPI imaging. We can also experimentally collect the ACMH loop from the particles. Because we collect the time domain response, dynamic response particles in the MPS system, thus we can experimentally collect the ACMH loop from the particles to compare with the DCMH loop of particles. We also can do the tumor imaging. We are building up the 3D imaging of MPI system. We can also do the tumor treatment by hyperthermia treatment. We also produce and calibrate the magnetic nanoparticles with varying shape, size, and element doping to vary the performance of these particles for different applications. So at the end of my talk, I want to acknowledge the funding source from the NIH and the TTU College of Engineering as well as the TTU STEM Center. I also want to acknowledge that most of the data I present here are produced by my students in my lab. Okay, that is the end of my talk. Thank you.
Excellent. Thank you very much, Kai. So we are tight on time, but we've got time for one or two quick questions. I should warn that most of the individuals here are not on the biomedical side of things, but there are a few quiet individuals that could maybe come off mute if they've got questions.
I have a small question, Kai.
Yes.
So looking at your imaging process, you use a DC field gradient, right?
Mm-hmm.
Now,
Yes.
wouldn't you see particle motion because of that? Wouldn't the particles move around because of the field?
Yes, that's a good question. So usually for the MPI imaging, we are waiting for particles to accumulate in the tumor region. So which means we will inject the particles, wait for particle to accumulate in the tumor region. And the other particles will be cleared away by the blood circulation process. So at that stage, the particle will be stuck inside the tumor cells. So even we apply the DC field, the particle are unable to move inside the tumor cells.
Okay, and a follow-up question to that is, since you actually have a whole setup which has a DC gradient field and everything else, can you actually guide these particles using magnetic field to the right location as well, or has that been explored?
Yes, so that is one very traditional method that the field guided the delivery of the nanoparticles. Is we cannot do that, but my lab we do not do that. Usually it's like after people inject the nanoparticle in the blood vein, they do not wait for the blood circulation to bring the particle to the tumor. They use magnetic field to guide them to the tumor region. So this is one application that people can lower the nanoparticle cost. So to make sure all the particles they accumulate in the tumor region, and also speed up the accumulation of particles to the tumor region in a short time.
Thanks, Kai.
Yeah.
Excellent. Any other quick questions? All right. Well, then, if there are no further questions, let's thank our speaker again. Much appreciated. Kai, well, we can talk offline if you are okay with posting some of this on YouTube. We'll just chat back and forth with email. Let's wrap it up there. Stop the recording. Thank you very much, everybody.
Thank you so much. Yes, thank you, everyone.